In 2024, Colossal Biosciences announced they had brought back direwolves from extinction. But how did they do that? Did they clone these animals using the same method they used in Jurassic Park? And if they did that, will we be able to bring back dinosaurs? Is it possible to clone a dinosaur? And how would you go about doing that? In 1993, Jurassic Park sold the world on a simple premise, an idea for how to bring dinosaurs back to life by cloning them from samples of ancient DNA. The idea seemed so plausible that actually inspired scientists to see if that could work. Unfortunately, the answer is no. But the story of why the Jurassic Park method doesn't work is also the story of what kinds of animals we can actually bring back from extinction and how. So let's do this properly. Let's look at the Jurassic Park premise, examine the three main reasons why it doesn't actually work, and then talk about the actual science of de-extinction, the kinds of animals scientists are already bringing back from extinction and how they're going about that. The main idea in Jurassic Park was cloning dinosaurs from samples of their ancient DNA. The premise was very elegant. All you need was dinosaur DNA, which you can find in a sample as small as a single drop of blood. But where do you get 100-million-year-old dinosaur blood? Well, we know there are mosquitoes from the Mesozoic era perfectly preserved in amber. Cloning a dinosaur, then, is a matter of finding a mosquito that had fed on dinosaur blood, a mosquito that had been perfectly preserved in amber for millions of years, extracting the DNA from that blood, filling in the missing pieces, and using some cutting-edge equipment to turn that DNA into a baby dinosaur. When the movie first came out, this didn't seem entirely absurd. Dolly the sheep was still a few years in the future, but people had cloned animals successfully in the lab before, and the science of studying ancient DNA was still in its infancy. The theory sounded convincing enough, and it seemed like something that might be just around the corner. But there are three fundamental problems with the Jurassic Park model, and the first of them is that DNA degrades. DNA is a molecule, and molecules don't last forever. When you look more closely at the double helix of DNA, you'll see that it's formed by a series of building blocks called nucleotides, and there are bonds between the nucleotides of the two strands that keep the strands together. In a living organism, these bonds are constantly breaking apart and getting put back together again. But after cell death, there are enzymes that break apart these bonds, and there is no mechanism for putting them back together once more. Microorganisms can also accelerate this process, and water is responsible for the majority of the damage through a process known as hydrolysis. By 2012, scientists could even tell how long it takes DNA to degrade. They were studying bone samples from an extinct giant bird from New Zealand, a bird called moa, and they were able to determine that the half-life of DNA is approximately 521 years. What this means is that after 521 years, the amount of DNA in a sample is reduced by half. After another 521 years, there's half of that, and on and on and on, until there's nothing left. Let's put that into perspective. The T-Rex lived in the Cretaceous period, that ended at about 65 million years ago. After such a long time, there wouldn't just be gaps in the DNA, there wouldn't be anything left. Even under the best possible conditions, all DNA in a sample would be gone after 6.8 million years. But you might ask, well, can't DNA be preserved? What about the amber they show in the movie? Well, it is possible to preserve DNA. In molecular biology and genetics labs, we keep DNA frozen. So in my lab, we keep our samples in a freezer at minus 20 degrees Celsius, and that should keep the samples fine for a few years. We also have access to a minus 80 degrees freezer and liquid nitrogen, which I think are standards for molecular biology and genetics labs, and these will preserve it for even longer. In fact, some of the best samples of ancient DNA we have, like a mammoth sample, are samples we found frozen. Compared to that, amber is simply not the best way to preserve DNA. It's very good at preserving physical structures, and we do have mosquitoes that date back to the Mesozoic era. We can see that their physical structure is perfectly preserved, but the DNA inside them is gone. Amber is subjected to temperature changes, to radiation, and it doesn't protect DNA from water, so hydrolysis is still going on and is still destroying DNA inside. There have been numerous rigorous studies trying to extract DNA from samples preserved in amber, and although some of these initial studies seem to have been successful, now they have largely been debunked, and what people believe is that there was contamination, so basically modern DNA had contaminated the samples and had been mistaken for ancient DNA. I did find one paper where they managed to successfully extract DNA from a sample preserved in copal, which is a precursor to amber, but even then, they said that the DNA, the concentration was too low and the material was too degraded to be of any use. The oldest sample of DNA ever recovered is a mammoth sample that is over 1 million years old, which is impressive, but is still over 64 million years short of T-Rex territory. The bottom line is that even if we found a mosquito that had bitten a dinosaur, the DNA inside it would be gone. But let's put that aside for a moment. Let's be generous and say, by some miracle of science or science fiction, that we did find a sample of DNA from the Mesozoic era. Even Jurassic Park concedes that material that old would be at least a little degraded, and we would need to fill in the gaps somehow. Their solution to that problem was filling in the missing sequences with sequences from frog DNA. There was a narrative reason for doing this. In Jurassic Park, the scientists understood the need to control reproduction in the park, which is why every animal in the park is female. Some frogs, however, are hermaphrodites, and by using frog sequences to fill in the gaps in dinosaur DNA, the scientists inserted this trait into their dinosaurs, which is why the animals were able to reproduce in the wild, and why Dr. Grant stumbles upon eggs at some point. This is important for the story. It's part of a bigger theme of Jurassic Park, the overpowering quality of nature and the impossibility of controlling life, no matter how advanced our science gets. However, regardless of the reproduction problem, frog DNA simply wasn't the best choice to do what they were trying to do. DNA isn't just a list of instructions. It's more like a recipe book where the instructions only make sense if you read them in the correct order, use the right ingredients, and make sure that every single part is combining in the proper order and in the proper way. Genes don't work in isolation. They interact with each other in complicated ways. There is a myriad of elements in DNA, enhancers, promoters, silencers, all working together to make sure things happen the way they're supposed to happen. Now imagine trying to rebuild this structure, trying to rebuild the cookbook by inserting pages randomly from another book entirely. A book that's written in a different language, a book about a different cuisine, a book from another planet. This is what it would be like to try to repair dinosaur DNA with sequences from amphibians. If you have to fill in the gaps, it would make a lot more sense to use DNA from a species that is more closely related to dinosaurs. The more closely related animals are, the more similar their DNA will be. If the animals are too far apart, it's possible that the more complex organism has genes and sequences that are entirely absent in the other. If we had to fill the gaps and sequences of Neanderthal DNA, for instance, it would make a lot more sense to use humans as opposed to fruit flies. For dinosaurs, at least some dinosaurs, the closest relatives are birds, so it would make a lot more sense to use that. Even then, however, patching dinosaur DNA would be an insurmountable problem. While birds and theropods share a common ancestor, they have been evolving separately for several million years. Gaps in dinosaur DNA wouldn't be empty, clean slots. They would be vast regions for which we have no map. Clearly, that's a major issue. But again, let's put it aside for the moment. Let's say we somehow were able to fill in the gaps in the DNA, or better yet, let's say that we were able to find a sample that is preserved enough that we don't actually have to fill any gaps. Would we be able to do it then? Well, there's another problem. The main strategy for cloning mammals is something called somatic cell nuclear transfer. I talked about this in a bit more detail in my video about cloning in Star Trek. So if you're curious to hear about that, do watch that video. But very briefly, to do this process, we would need 3 things. We would need DNA from the animal that we're trying to clone, in this case a dinosaur. We would need an egg cell from a donor, in which we could implant this DNA. And we would need a surrogate. In the case of mammals, we need to implant it into the womb. In the case of animals that lay eggs, we would at the very least need a surrogate egg. So let's go through these in stages. First, the DNA. So actually, to clone an animal, we don't use the DNA in isolation, we need a nucleus. The nucleus is the central part of the cell, and it's where the DNA is, separated by a membrane. To clone a T-Rex, we would need a living T-Rex cell from which we could extract the nucleus. Obviously, we don't have that. The second element we would need is an egg cell that usually comes from a donor. Now, when we do cloning, like when we clone sheep, we usually use a donor of the same species for obvious reasons. The cell provides a lot of the things for the embryo, but the cell is adapted to a nucleus of its own species. We would need a living T-Rex egg cell, which we don't have. An alternative would be to use an egg cell from a closely related species. So maybe we could take an egg cell from a bird, maybe an ostrich, because it's the largest bird we had, but still, the internal environment of that cell is adapted to ostrich DNA. There's no guarantee that any of it would work with the DNA of a dinosaur instead. Third, and most importantly, we would need a surrogate. Again, in this case, let's assume we will use an ostrich egg. Again, there's no guarantee that it would work. First of all, the size is wrong. Then the internal environment of the egg is adapted for an ostrich. We have no idea if the conditions would be suitable for a T-Rex, for instance. An ostrich egg hatches in 42 days. How long does a T-Rex egg take to hatch? We don't know. Was the shell thicker? We have absolutely no idea. We lack information, and there's no reason to think that it might work. And that's only considering one species. Different dinosaur species had different eggs. There were species that had soft shell, leathery eggs, for which we have absolutely no equivalent today. Without a living T-Rex to provide an egg, there is simply no biological path through which we can make this work. DNA alone, even if it was available, is insufficient, at least with the technology we currently have. Despite these issues, though, de-extinction is a living branch of science. Scientists are looking at species that disappeared in the last few hundred or few thousand years, from which we can get usable DNA. After all, DNA was extracted from an extinct species for the first time all the way back in the 80s, when a group from the University of California, Berkeley, extracted quagga DNA from a dried muscle tissue in a museum piece. These projects are all about ecological restoration and advancing conservation tech. And the company leading the charge is one called Colossal Biosciences. Their flagship effort is the woolly mammoth project. Mammoths went extinct at about 4,000 years ago, and we have excellent DNA samples from frozen specimens. Naturally, we don't have any living cells, so cloning is not possible, but we do have a close living relative, the Asian elephant. The way they do this is they start by sequencing the DNA of both animals, the ancient DNA from the mammoth and the DNA of an Asian elephant. Then they compare these sequences and study the mammoth DNA, looking for cold weather adaptations, for sequences and genes that are different and that make the animal specifically suited to cold weather. And these would be things like shaggy hair, curved tusks, different fat deposits, and a different type of hemoglobin, things like that. For this project specifically, they have already identified 65 genes. The next step is to edit the DNA of an Asian elephant using CRISPR-Cas9 to introduce the changes that will make it more like a mammoth. This is exactly the same process this company used to bring back direwolves in 2024. In October of that year, they announced the birth of Romulus and Remus, two direwolves, according to them, that have been named after the legendary founders of Rome. A little later, they also got a little sister, which they named Khaleesi. And these animals are now alive and well in an undisclosed location. But at the time they were announced, this was a big thing. They made the cover of Time. I think they invited George Martin to see them. They even had a picture on the HBO prop of the Iron Throne. But shortly after all that noise, the company was heavily criticized, with several scientists saying those animals were not actually direwolves. And they were correct. You see, the process to create the direwolves was essentially the same that they are now using to create mammoths. But as you can see, they are not actually cloning an animal or generating an animal from the sample of ancient DNA. What they are doing is modifying the DNA of a living animal. In the case of the direwolves, they used gray wolves. So what we have there is a genetically modified gray wolf, and I think in that case they changed 12 or 13 genes. The same will be true of the mammoth when it happens. It will be a genetically modified Asian elephant, and it will be different from mammoths in ways we won't even be able to tell, because no one knows what a mammoth was actually like. But acknowledging this doesn't diminish the discovery in any way. Proponents of these ideas say that restoring mammals to the Arctic, for instance, could bring back grasslands that could contribute positively to the fight against climate change. People that speak against the theory say that these efforts, that we don't know how it will affect the ecosystem to bring back an animal that was extinct thousands of years ago through something other than human activity, and that these kinds of efforts take resources and attention away from the effort of actually keeping endangered animals alive while they're still here. In order to properly bring back an animal from extinction, at least according to the technology we have now, we would need to clone these animals, so we would need living cells. Since we know these now, we have frozen zoos, which are facilities that store material, that freeze material from endangered species to give us choices if these species should become extinct. In the early 2000s, they tried to bring back a type of wild goat named bucardo that went extinct. They collected material from the last individual, a female nicknamed Celia, in 1999, and she passed away in the year 2000. Because the animal is extinct, they needed to use an egg cell from a goat and a surrogate goat as well to grow the babies. And they used domestic goats and also some hybrids with a type of Spanish goat. And because the process has a low chance of being successful, they fertilized 154 eggs, which they then implanted on 44 females. Of those, only 7 became pregnant and 6 of them miscarried early on. So only 1 managed to carry the baby bucardo to term. But when the animal was born, it died hours after birth due to respiratory complications because it had a malformed lung. And these kinds of congenital malformations are very common in cloned animals. So the process is imperfect, and even freezing material from animals now is by no means a guarantee that we will be able to restore biodiversity. So, can we clone a T-Rex? Unfortunately, the answer is definitively not. The DNA is gone, we have no frame of reference, and the biological infrastructure to do something like that is long gone. We can, however, try to bring back animals that have not been gone for that long, and we can create functional proxies for animals that were extinct long ago. We might never be able to walk with dinosaurs, but we will be able to walk with mammoths, dodos, and direwolves, or at least a close enough relative. And that's not a bad consolation prize. I know we focused mostly on the cloning of animals and cloning from ancient DNA samples on this video, but if you're also interested about human cloning, then by all means watch my previous video, which is about human cloning in Star Trek and in science fiction in general. For now, this is it. I hope you enjoy this. Thank you for watching, and until next time, live long and prosper.